Alright guys, so this lesson is going to be our last one for our genetics unit, and it's all about the fruit fly. So just to kind of give you a little bit of a background, so we've talked a lot about Gregor Mendel and Mendelian genetics, and we've looked at his experiments and how he kind of gave us the basics in terms of dominant and recessive traits, but we know that not all things fit Mendelian ratios, and so another very important area of genetic study was the fruit fly. And actually, in a lot of college labs, if you are taking either biology or it could just be in genetics, you may even actually do the fruit fly experiment. And so we're just going to take a look at fruit flies and why they're useful and things that we've learned from fruit flies. We're not actually going to do the fruit fly experiment, but I do want to at least tell you a little bit about it. And so peas are to Mendel as fruit flies are to Thomas Hunt Morgan. And so this is the guy that's responsible for a lot of other material that we know about genetics by studying fruit flies. And so it was through the work of Thomas Hunt Morgan that we first associated pretty much the loci of certain genes, knowing that that gene is in a particular location on a particular chromosome. And this is the scientific name of the fruit fly, Drosophila melanogaster, and this is specifically the type of fruit fly that he used for all of the experiments. And so why the fruit fly? Of all the things that we could look at, why the fruit fly? Well, we're going to take a look at that they're easy to cross, they don't have a lot of chromosomes, and so it was from all of these fruit fly studies that we get a lot of our non-Mendelian inheritance. So things like the specific loci, and that we may not always have independent assortment. According to Mendel, the genes were separated during gamete formation, but we know that if genes are on the same chromosome, they might be inherited together, and that's what we call linked genes. The concept of sex-linked traits, like we looked at in our last lesson. The concept of epistasis, and you have an example of this in your problem packet with the labs, how that some genes can affect the expression of other genes. That concept of gene mapping, where we looked at the crossover rates, pretty much the higher the crossover percentage and recombination percentage, the further apart those genes must be. The multiple allele concept, that there could be more than just two possible alleles. I told you with Mendel's experiments that he got lucky. Every single one of those P traits were single gene, two alleles, dominant, recessive. And we know that not all inheritance is going to follow that pattern. And so you could have more than two possible alleles for a trait, which means more genotypes and more phenotypes. And as I mentioned earlier, just that specific gene location. And so the fruit fly was an easy specimen to use and study because they are very common. They're harmless. They don't really cause disease or anything like that. They're incredibly tiny, which kind of can make them a little difficult to work with, but they make babies like crazy. Okay, they are prolific breeders, and so they produce hundreds of offspring in one mating, and it only takes about 2 weeks to create some fruit fly babies. And so you can study them in a lab in a relatively short period of time. They also only have 4 pairs of chromosomes, so it makes them a lot easier to study those chromosomes under the microscope and to look at the differences amongst the chromosomes. And so just to kind of give you some background info on the fruit fly, pretty much the purpose of fruit fly experiments, now this is going to seem weird to us now because we already know the inheritance of many of these traits, but the purpose of a fruit fly experiment is to try to determine the inheritance of certain traits. Like, are they Mendelian, dominant, recessive? Is the trait linked to another gene? Are they sex-linked? Are they autosomal? Are they dominant? Are they recessive? There's so many different types of inheritance, and the purpose of a fruit fly experiment is to try to determine how are those traits inherited. And so essentially we kind of have two basic types of traits. We have what we call the wild type fruit fly. And this is, think of it like your cookie cutter standard fly, okay? Your basic fruit fly. Some examples of your wild type are red eyes, normal wings, a yellowish brown body. Now, for each of your phenotypes, we have what we call mutant phenotypes, which is anything other than wild, okay? So wild type is kind of like your standard fly, and anything outside of that wild type we consider a mutant. And so this could be white eyes or sepia eyes or purple eyes. It could be no eyes. There's different types of wings. There's different colors of body. There's the wild type when it comes to bristles and multiple types of traits. So pretty much you always have your standard fly, and then you have what we call our mutant phenotypes, which are all of the other possibilities for that trait. And so the purpose behind a fruit fly experiment is to try to determine how is a particular mutant inherited in relation to the wild type. And so essentially what you do with fruit flies is you cross them, you look at the babies, you count the babies, you look at their phenotypes, and then you try to determine how that trait is inherited. So these are some examples. So here's my wild eye, and then you have eyeless, bar eye, lozenge eye, lobe eye. So there's an eye shape, the bristles on the little head of the fly, the wing shape, the eye color. So pretty much for every trait on the fruit fly, you have your wild and then you have your mutant. And so because when you're going into a fruit fly experiment, you technically don't know how that trait is inherited. It's your job to count flies, look at the ratios, and from the ratios determine the inheritance pattern. So even though you could Google it and find it online, if you were to do the fruit fly experiment, it is your job to figure out how a particular trait is inherited. And so going into this, we don't necessarily know what's dominant and what's recessive. And so when it comes to doing genotypes for fruit fly traits, we're going to write them a little bit differently. Now, you are not going to be doing fruit fly Punnett squares, and I'm actually taking away the fruit fly chi-square problems, but I do need you to know how to kind of write out fruit fly genotypes. And so once again, this is without knowledge of dominant and recessive. So the way you do it is you always use the letter for the mutant phenotype. So wild type is always crossed with a mutant phenotype. That could be anything, any trait, any phenotype that is not the wild, okay? So for instance, we could be doing purple eyes, okay? We could cross a purple-eyed fly with a wild type red-eyed fly. And so you always use the letter for the mutant. So if we were doing purple eye, we could just use letter P for purple, okay? Then if it's a wild type gene, you put a superscript of a plus sign. So we always denote either just P would be the allele for purple, and then P plus would be the allele for wild, okay? So once again, we're no longer using capital lowercase because technically we do not know the inheritance. We don't know if it's sex-linked, we don't know if it's autosomal, we don't know what's dominant, what's recessive, okay? So here's a couple examples. So you have, in this case, we're using vestigial wings, which vestigial wings are kind of these little shriveled up kind of wings. And so if you had a homozygous wild, there's my letters for vestigial, but notice it's plus plus, okay? So that tells me I have two wild type alleles. Homozygous vestigial would just be the VG for vestigial. And if it's heterozygous, notice you have a plus on one and then just a VG on the other. Okay, so if we were doing this for the purple eye, you could have a homozygous purple, which would be two P's. You could have a homozygous wild type, which would be P plus P plus. Or you could have a heterozygote, which is P plus P. Okay, so this represents the wild type allele, and this represents the mutant allele. We also have to look at males versus females because we don't know if the trait is autosomal or sex-linked. We have to be able to sex the flies, which means we have to identify whether they're male or female. When you're counting your fly offspring, you have to separate them because that is what's going to tell you whether or not it's autosomal or sex-linked. Remember, if it's autosomal, you should not see a significant difference between male and females in who has it, who doesn't, who's a carrier, anything like that. But if it's sex-linked, you could see a distinct difference between male and female inheritance. And so the way you identify the males versus females, there's a couple of different ways. I actually had to do this experiment in my genetics class in college, And to me, the easiest way to tell the difference between male and female is the black butt. Now, I know it's technically not a butt on a fruit fly, but pretty much to us that would be their backside. So you can see that the male is a lot rounder and it's got a thicker black band there. It's darker. The female has more of a pointed back end and it's not as dark. Okay, the female is also a little bit bigger than the male. Now, something else to look at, and this is really hard to see without a really like a good microscope or a good magnifying glass, but males have sex combs, and so on their kind of front legs here, they have these little bristles. They're kind of hair-like looking structures, but your sex combs, it actually helps the male kind of mount the female, if you will, whenever they are mating. So you do have to be able to tell. If you were to do this experiment in class, you would have to individually identify each fly as either male or female, along with whatever phenotype they had for the trait that you are crossing. Now, one of the things that makes fruit flies so easy to work with is their life cycle. Okay, so it takes about 10 days at your room temperature. If it's colder, it takes a little longer. If it's warmer, it may go slightly faster, but it's normally within a couple of weeks, like I mentioned earlier, you're going to have fly babies. And so what you do is you literally order flies. You order the genotypes that you want to cross. You order males of one type and females of another type. And then you cross them and you look at the outcomes. So the fruit fly experiment basically involves crossing the phenotypes that you want to cross and then identifying the offspring and counting them. Now, this is one thing that does overlap with Mendel's work, is we still have that same basic experiment, in that you start off with the P generation. They are homozygous of opposite trait. It's usually a homozygous wild crossed with a homozygous mutant, whatever that trait you want to cross. You take your F1 babies, which are going to be hybrids or heterozygotes. You look at the outcome, and then you let the F1s mate, and they produce what we call the F2s, and you look at the traits there. Okay, and it's from those results, by looking at the numbers and the phenotypes that you get in the F1 and the F2, that you can then try to determine the inheritance of that mutant trait. And so this is what Thomas Hunt Morgan did. He and his crew, he and his group that they worked together, they would cross a wild type with a mutant. So this is always going to be your P generation. It's always going to be a homozygous wild crossed with a homozygous mutant. They would look at the F1s, they would let the F1s mate, and then they would produce the F2s. And from that, they would look at the ratios. Now, they might get Mendelian type ratios, and if that's the case, that would tell them that it's autosomal. You have a basic dominant recessive pattern. Or if they didn't get Mendelian type numbers, that would help them determine if it was something like sex linked, or maybe they were linked genes. And so anytime those results didn't match, it would lead them to other types of inheritance. And so the purpose of the fruit fly lab is to kind of take these Mendelian ratios, form a hypothesis for how we think a particular trait is inherited, and then we're going to use a chi-square analysis test to analyze our results. And so we're going to take a look at chi-square more closely tomorrow. It is like a mathematical formula. If you're in statistics class, you may or may not have done chi-square yet. But essentially, the purpose of the fruit fly lab is to determine the inheritance of a mutant phenotype. Now, once again, you could easily Google and find out on the internet how purple eye is inherited, or how sepia eye is inherited, or how vestigial is inherited. But the whole purpose of this experiment is to really test your genetic knowledge and to look at ratios and be able to apply what you know to determine a particular inheritance pattern. Now, once you determine that inheritance pattern, you take your expected ratios and you apply what you actually get in real life to what we should have gotten based on probability, and that's where the chi-square analysis comes in. And so the purpose of a chi-square analysis test is what we call a goodness of fit test. It's essentially how close is our actual data to what we should have gotten based on probability. Okay, So it always compares what we observed in real life to what we expected based on probability. And when we take a look at the formula, you're going to see that we always have O's and E's. We have the observed values, what we really got, and we have the expected values, what we should have gotten, okay? So what chi-square does is it determines if that's a good fit, okay? So let me kind of give you an example. Let's say we have, this is not just applying to genetics, okay? We could apply this to any kind of probable situation. Let's go back to our good old classic coin flip, all right? So if I flip a coin, what I would expect is to get 50% heads and 50% tails, okay? So let's say that I flip a coin 100 times. Well, what if I get 60 heads and 40 tails, okay? Now, obviously my expectation is that I would get 50-50. So if I get 60-40, how close are those numbers to the 50-50 that I should have actually gotten? And so what chi-square does is it's actually a mathematical way for us to kind of analyze what we got to what we should have gotten. And so if those numbers are too far away, then it would probably lead us to say, well, that really wasn't a fair coin, or there was something wrong with that coin. Or if the numbers are close enough, it'll kind of help us decide, well, yeah, that was a fair coin. You know, we aren't going to get perfect results every time. So what we're using chi-square for is essentially, it's a mathematical way to determine that our numbers are close enough to the expected, or they're not close enough to expected, okay? It's just a way to kind of analyze that using math. Now, we talked about a null hypothesis pretty early in the year, and we learned that a null hypothesis pretty much says that there is no relationship between the independent variable and the dependent variable. And so that's kind of the same thing that we're looking at here. So our null hypothesis says there is no significant difference between what we got and what we should have gotten. So based on the chi-square test, what we're going to do is we're either going to accept this, okay? If we accept this hypothesis, that means that our numbers, our ratios were close enough to the expected ratio, and therefore we're essentially going to accept that that is the inheritance pattern, okay? Or if we reject this hypothesis, we're saying there is a significant difference, and therefore that must not be our inheritance pattern. Okay, so what you would do if you had done the fruit fly experiment is you would have counted all of these flies. You would have come up with ratios that would indicate a particular inheritance pattern. And then we would use those observed numbers and compare them to what we should have gotten if it happened perfectly. And when we would either accept this hypothesis and say that we were right, essentially, that that is the inheritance pattern, or we would reject this hypothesis based on the chi-square analysis, okay? So accepting means that any differences are just due to simple chance, because we know that reality is not going to perfectly match the expected, okay? If we reject it, it means that the difference is big enough that it must be due to other factors, okay? So now let's take a look at the chi-square formula, okay? So this is the chi, like the Greek letter, and you literally are solving for chi-squared, okay? Never are you taking the square root of anything. So when you come up with your answer, you're literally saying chi-squared equals, okay? And you're going to come up with a number here, okay? So this is the formula that we're going to use to solve for chi-squared. So this big Greek E looking symbol here, okay, I don't know the name of that, but it means the sum of, okay, so we're going to sum up or add up the observed minus expected squared divided by the expected, okay. Now the reason we're adding it together is because we're going to have multiple expectations, okay. So if we were doing the heads and the tails when we're flipping a coin, there's two outcomes. There's how many heads did you get and how many tails did you get. So you take the observed, what you actually got, and you subtract the expected. So if we wanted to plug in the formula for that problem that I mentioned earlier, what we would do is say, okay, I observed 60 heads. So 60 would be my observed. My expected would have been 50. So 60 minus 50 squared divided by 50. And then I would add that, this is where the sum of comes in, okay, to my tails. So 40 minus 50 squared divided by 50. And that's how you fill out your chi-square formula. Tomorrow we're going to take a look at some actual practice problems, and you're going to get more familiar with this. Now, in statistics, and I will admit I'm not a mathematician. I loved math. I always thought it was fun. I enjoyed it. And back when I was in math classes, I understood it. I have been out of math classes for a very long time. But in statistics, if you've ever taken it, or if you're taking it now, what you'll find is that there are usually a lot of tables. Like you use calculations and then you compare your data to a table. These tables have already been created by these mathematicians, and so we're just going to use it, okay? It's based on a probability table that we are able to determine whether we accept or reject, okay? Now, honestly, I can't really tell you the whys. I can just tell you this is what you do. This is how you do it, okay? So when you're using a chi-square table, it's based on degrees of freedom, and this is however many categories you had minus 1. So if we were doing the coin flip, I had two outcomes. I had heads or I had tails. So if I were looking at my chi-square value on the table, it would be degrees of freedom of 1. Okay. If I have two phenotypes, the flowers were purple or they were white, I would have a degrees of freedom of 1. If I were doing a dihybrid cross, I have 4 different phenotypes, so I would have a degrees of freedom of 3. It's always however many phenotypes you have minus 1, or however many categories you have minus 1. Now, the critical p-value is the number that you use to decide if we're going to accept or reject, okay? And what the critical p-value is showing us, it's saying that these are the values that we would expect to get a certain percentage of the time, okay? So it represents the percent that your results are due to chance. So the higher that percentage is, that means, yeah, it's probably just a random, just a chance thing. And so the higher your percentage is, then we're going to accept, okay? If it's a really, really low percentage that's due to chance, that is when we're going to reject. And so the cutoff is always 0.05, okay, which this means 5%. So if we would expect results like that 5% of the time or greater, we're going to accept. If we would expect those results less than 5% of the time, then we're going to reject. Okay, now I know this may sound kind of weird, especially since you can't really see a table. So let's take a look at an example of a chi-square table. All right, so over here is my degrees of freedom, okay? And then up here are my p-values, okay? These represent percentages. So this is telling me that these are the numbers that I would expect 95% of the time. These are the numbers I would expect 90%, 80%, 70%, all the way down, okay? Now, once again, the 0.05 is my critical p-value, okay? I'm not sure why that's highlighting all of that over there. So you can see this right here is where I kind of divide my table. Anything less than 0.5, okay, this means there is a significant difference, okay? That means that these numbers here, I would expect to get less than 5% of the time. And this is when I am going to reject, okay? I'm going to reject my null hypothesis. I do not like this. I don't know why it's doing that. Sorry guys, let me switch to a pen. So everything on this side of the table is when I'm going to reject. Because what that means is that I would expect to get those results less than 5% of the time, okay? And that means there must be something else going on. This is not very common, and what I want you to notice is how big these numbers are, okay? All of these numbers in this portion of the table All right, all of these represent the chi-square values. So when you solve for chi-square equals, okay, you're going to get a number here. These are the numbers that you're looking at in the table. Okay, these represent chi-square values. And so if we would expect to get that less than 5% of the time, we're going to reject. Now, anything on this side of the table, that means we would expect to get that more than 5%. It could be 10%, 50%. And so this is when I'm going to accept, I don't know what is going on. This is when I'm going to accept my null hypothesis, okay? And what we're saying is there is no significant difference. So what we are testing is close enough to what we should have gotten, and we're going to accept. We're going to accept that that is a fair coin. We're going to accept that that is our inheritance pattern, okay? So let me just kind of give you an example. Let's say that our chi-square value, let's say we did the coin toss. I'm not sure what the number we actually would get if we did a 60-40, so I'm just going to completely make up a number here. But let's say that I got a chi-square value of 1.75. Okay, so I'm just going to make up a number here. I got a chi-square value of 1.75. So if I was doing a coin flip, then I would use 1 degree of freedom. Okay, I'm going to go back to my highlighter here. I would use 1 degree of freedom. And what I'm going to do is I'm going to look up 1.75 on my table, and I'm going to see where that fits. So 1.75 would be here. Okay, it would be between the 1.64 and the 2.71. So what that means is I would expect to get those results between 10% and 20% of the time. This is going to be in my accept range, so I would accept that chi-square value. That means there is no significant difference. So if I got an accept for the coin flip, we're just saying, yeah, that's a fair coin. That falls within a normal range. Okay, if I got a number over here on this side of the table, that is when I would reject. Okay, so anything 3.84 and higher, I'm going to reject that. All right, so I know that may not make a whole lot of sense. I'm hoping it'll kind of come together tomorrow when we take a look at some practice problems. We are going to be doing some math tomorrow. And so what I need you to do, and hopefully you can do this in class today, is this is just an overview. Since we aren't actually doing the fruit fly experiment, we're not doing the real one and we're not doing the virtual one. The virtual one is no longer available. There's a video. It's very short. It's only 5 minutes. This is by Bozeman Science. And what he's doing is he's explaining the virtual fruit fly experiment, okay? And he's kind of going over a little bit about the chi-square test. Now, there's also a Bozeman video over the chi-square test, and I should have added that here before I posted this. I didn't think about it. I'm going to send that link to Miss Petty. And so I would encourage you to not only watch the fruit fly, hopefully you're going to watch it in class today, but I would also encourage you to watch the chi-square. I'm going to explain it to you tomorrow on the video. We're going to do some practice problems, and then you're also going to be expected to do this on the test. So I would encourage you to maybe watch the video if you need help. All right, well, that's all I've got for today. I will be back with you tomorrow teaching you how to do chi-square, the actual math formula part of it. And I hope you guys have a good day.